Welcome back to Lecture 2 in the Neuroeconomic Methods course. And as you might guess, this lecture focuses on fMRI. So what we'll do here is we'll briefly talk about how fMRI works, so how the MRI scanner works, how functional magnetic resonance imaging works, and then we'll talk about something that's very relevant to your labs, which is how we do pre-processing, why and how we do those steps. So let's start with fMRI. So this is a technique that was developed in the early 90s, basically to study brain activations. And one of the cool things is you can do this simultaneously while people in the scanner are doing a task. So we can look at relatively complex decision-making tasks while participants are inside the scanner, which is very cool. So we have this kind of process here. So we have a mental task that a participant is performing inside the scanner. This then we know activates neurons that are involved in this task. And what we're actually measuring is the bolt response, which is related to when neurons are active, because neurons require energy, and one of the types of molecules that supplies this energy is oxygen. And this, as we'll see in a minute, then constitutes the bolt response. When the brain sends more oxygen to regions that are active, we can see an increase in the bolt response or increase in MR signal intensity, which you can then map using statistical analyses at a later point. Here's an example MR scanner. Typically, I take you to the MR site, but given the current situation, that's unfortunately not possible. So how does fMRI work? Well, let's first talk about how MRI works. So how do we make structural images of the brain or any type of tissue in the scanner? And that's one of the cool things about scanners. They're actually highly flexible machines, and they allow us not to just make images of brains, but also any other body part in action. So you could also look at hearts while they're moving or lungs while they're breathing. So this is one of the cool things that we can look at all sorts of tissue types, all sorts of body parts, and assess them using MR. So you can make clinical assessments, but that's not really what we're interested in. We're much more interested in another sort of level of versatility, namely functional changes in blood flow to different regions that are active during specific tasks, such as economic decisions. And that's also what MRI technology allows us to do. So we typically, and in the neuroeconomics course you already saw this, we talk about these activations on the brain here in the amygdala, and then we can talk about activation differences across different conditions, which allows to make inferences about how the brain works, how different parts of the brain are active or responsible for different types of cognitive and affective processes. Well, the MR machine is actually even more flexible than this, and we can make such diffusion tensor images, and there are many other types of sequences that we can use to image different things, different aspects of brain anatomy. This is one aspect of it. So in addition to looking at the anatomy of the brain using these simple anatomical or T1 images, as you'll see in a second, we can also look at how water diffuses in different directions within the brain. So we have from front to back or back to front, we have this diffusion in this dimension here in green, from bottom to top, so superior to inferior, or the other way around. We have diffusion in blue, and from left to right, that is shown in red. And this tells us basically how different neuronal areas are interconnected within the brain. And you can even compute or estimate the connection strength using probabilistic tractography on these types of images that give you an understanding of how thick or how dense the connections are between different neuronal clusters. And that's something that you can correlate with behavior in other settings. So it's about the connectivity between different areas when it comes to diffusion tensor imaging. We can also assess connectivity using functional brain images, but we'll talk about this at a later point. My point here is that this is a heavily versatile method that we can use for many, many different things. So let's have a look at how the MRI machine works. So MRI stands for magnetic radio imaging, or radio frequency imaging. So at the basic level, we have a very strong magnet that with most magnets or with most machines nowadays is about 3 Tesla, which is many thousand times the magnetic strength of the Earth, of the planet. So when you place a person or tissue inside a scanner, inside a magnet that has such a strong magnetic field that is also static, what will happen is the following. You will find that atoms that are susceptible to magnetic fields, such as hydrogen atoms, which in daily life basically spin around in random directions, right? Atoms have a spin, and these directions are relatively random in daily life, with no net direction across all of your different hydrogen atoms. And that's obviously, hydrogen atoms are parts of water molecules, of which the body has about 70%. So 70% of the body is made up of water. So we have a lot of these hydrogen atoms, and they spin in random directions. Now, when you place a person in a scanner, then what happens is these hydrogen atoms align with the magnetic field. So if the direction of the magnetic field is upward, in the scanner it's actually horizontal, then what you'll find is that these hydrogen atoms within this magnetic field align in the direction of the magnetic field. Most of them will align in the actual direction, and some of them will align in the opposite direction, so with a 180-degree flip. But they will generally be aligned with the magnetic field, leading to a net magnetization. So the average magnetization of these hydrogen atoms will be parallel to the magnetic field. Now, we can play with this. By placing a person in a scanner, we have a net magnetization that is parallel to the scanner's magnetic field. And we can disturb this by sending in a radio frequency pulse, which is basically adding energy to these atoms. And this will throw them off. If you have a specific frequency, this will throw them off their magnetization with the magnetic field and flip them by about 90 degrees. So they're no longer spinning in this direction. They're now spinning perpendicular to the magnetic field. But they're carrying all this energy from the radio frequency pulse. So as soon as you turn off the radio frequency pulse, they will actually want to go back and realign with the magnetic field to go back to where they were before. While they're doing this, while they're spinning back to realign with the magnetic field, they're releasing the energy that was put into them via the radio frequency pulse. And if you then use a receiver coil, that's the imaging part here, we can pick up the radio frequency energy that is emitted by the hydrogen atoms that are going back to their sort of natural state within the magnet. While they're doing that, they're emitting the energy, and you can pick up this via a radio frequency coil that's also inside the scanner. So it's an electromagnetic radiation that's emitted by these hydrogen atoms going back to their aligned state within the magnetic field. So this is basically very superficially how the scanner works. So what is nice about this method is that different brain structures have different hydrogen concentrations. So this means that a tissue type that has more hydrogen atoms actually absorbs more energy, and it will take longer for this tissue to go back to the original state and release this energy. And this difference in hydrogen concentrations is something that we can image with the MRI scanner. And this is an illustration of how this is done. So tissues containing more water absorb more energy and recover more slowly. And that's what's shown here in the red, right? So this is a tissue type that has more water, and in the blue we have a tissue type that has less water. And the goal of structural imaging of the T1 contrast is basically to find a time point where we maximize this difference between these two tissue types. And that is basically where we want to take our measurement and then start the next measurement, basically, right? So this difference is due, again, to the proton density of hydrogen atoms within these tissue types. And what this T1 contrast then measures is the recovery of net magnetization in the longitudinal direction. And that means in terms of the alignment with the scanner. The TR here is the time point where we take a new measurement. So we start a new pulse. The TR is the repetition time, or the interval between two successive excitation pulses, if you will, but they're these radio frequency pulses that we talked about earlier. And so if we measure at about this time point here, we find, so just before the new TR, we find that we have a maximum difference between these two tissue types, and therefore maximize the contrast in the image that is recorded by the scanner. Here's another illustration of this, and basically with tissue types that have short T1 recovery rates and tissue types that have long T1 recovery rates, those are the ones that contain more water, and those are the ones that contain less water. And you can see that there is a difference in recovery time between gray matter, which takes a little bit longer and therefore has more water, and white matter, which takes a little bit less long. And obviously we have different types of tissues that we can then picture. And obviously CSF, which is a fluid that is within the brain, within the ventricles of the brain mostly, has the longest T1 recovery time. And this is something that will then appear in black. This is something that will appear in relatively light gray, and this is something that will appear in relatively dark gray in T1 images. So this is the method that we use to make structural images of the brain, high-resolution structural images of the brain, and the basis of the T1 contrast, which is one of the main images that we make using the MRI scanner.